The universal principle of being conscious of what you put in your body at the simplest level.
Martha, welcome to the podcast.
Thanks for having me, Nick.
Yes, we were just chatting offline about all of the topics that you've studied and are involved with. We're today going to cover some things such as lipopolysaccharides, glyphosate, the microbiome, Parkinson's, and the interconnection between a lot of fields that are traditionally not seen as interconnected. So before we begin, how did you get involved in all this? Because your story is an interesting one in itself.
So my actual training and background out of university, I was trained as an accountant. And of course, in accounting, everything must balance. It's nice and tidy. And coming out of that, I went to work for one of the large public accounting firms as an auditor. And they train you there to never take anything at face value, examine the evidence for yourself. So that kind of mindset and training sets you up to be a different kind of person in the world. And then they trained me on a way to look at a business to identify business risk called transaction flow review, which is where you draw these big flow charts of everything going through the business. And you're looking for those breakpoints. And fast forward from that point, in 2002, my 44-year-old healthy marathon running, looked perfect husband was diagnosed with Parkinson's. And I was like, that's an old person's disease. And the way they hand you the diagnosis is it's like this heavy weight that gets dropped on you with really no hope. And from my kind of mental thinking of never take anything at face value, examine the evidence for yourself, I started down this path of examining the evidence for myself. And with that, mapping out the risk, like what's flowing through the body. So the first place I started was, okay, I got to look at the food because that's the main thing that's flowing through the body. And so I spent about a decade looking at our food supply, changing what we were eating, trying to go all organic, which there wasn't a lot of available back in 2002. And then saying, okay, well, if I can't find organic, can I understand like what we're doing to our food supply? Like what kind of chemicals are we using? What is this thing called genetic engineering? How does it work? Could it transfer to us? And if it could transfer to us, like what might that mechanism be? So I set up kind of all these questions along the process of looking at food and studying food science and nutritional epigenetics, even like how different foods affect gene expression in our body. And then in 2014, I read a book by Dr. Martin Blaser called Missing Microbes. And Dr. Blaser is a world-renowned infectious disease doctor, spent a lot of his career researching H. pylori that is connected with ulcers. And once that was discovered, of course, everybody was given antibiotics. And his book, Missing Microbes, was actually about us growing up in the age of antibiotics, which for the last, let's say 60 years or maybe a little longer now, we've all had a massive amount of antibiotics. And what he was studying was the connection to the rise in what are called non-communicable diseases or chronic diseases, as opposed to acute infection. And through that work, basically started to show that we had depleted our microbiome. And I had never heard this term microbiome. Like, what is that? Well, it's the trillions of bacteria, fungi, and viruses that live in and on our body, and they function as our internal pharmacy. And we are about 2/3 microbial in terms of cells. So we're kind of like a giant petri dish, really. And so that was just this really big epiphany for me. And about 6 months later, I read the first paper where they showed the two different types of Parkinson's. One is primarily tremor dominant, and the other is posture and gait dominant. And they could divide those people by the bacteria in their gut. And it was like, home run. The gut is the general ledger, sort of back to my accounting. That's where it all balances out, and the record of your history is there. And so I actually quit my job that December and started funding some research at the University of Chicago that introduced me to Dr. Jack Gilbert, one of the pioneers in the microbiome space, who was later my co-founder along with Dr. Suzanne Vernon of the Biocollective, where, I had this idea, okay, we're going to collect people's poop because that's how you look at the microbiome. And people were like, what are you, nuts? And I was like, no. And not only are we going to collect people's poop, because there was a project going called American Gut and a company called Ubiome, and they were basically just taking a little swab. And we actually were like, no, you need the whole thing. So we designed this kit. People look at us like we were crazy to send you a box, a giant box. It said revolutionary on the side. And it had this little hammock, and you went in the hammock and this picked up the arms of the hammock and stuck it back in the cooler with this big freezer brick and, put your sticker on there that said, I did my duty. And you shipped it back to us. And we then, inside the bag, so it wouldn't be exposed to oxygen, would basically homogenize the sample and then squeeze it into a syringe and put it into these little 1 gram aliquots that we made available to scientists all over the globe. And then we did we sent them off for sequencing ourselves and started building this big data bank to try to kind of comb through all the data and start to see not just Parkinson's, but across the population, like what are the things that are disrupted in our system.
And so what did you find?
We label all these different diseases, but there are some very key metabolic disruptions that are shared across diseases in the microbiome that are rarely connected, because when people study it, they're still studying it in a very reductionist way. And what was one of the really interesting things to me that we found, so we had our Parkinson's cohort, and we actually got hired by Philip Morris, the tobacco company, to look at microbiomes of smokers that we had. And so we were doing a bunch of analytics on that. And, some people will know this, but not many. Parkinson's is less prevalent in smokers. And as we were looking at the microbiome data, we could actually see kind of the flip metabolic profile of different microbes and the metabolites that they were producing in the smoker and the Parkinson's. But another kind of really crazy other thing that we found is the people in our lab, after a year or two of collecting samples, they came to me and they said, we can tell if somebody has Parkinson's just by looking at their sample when it arrives in the lab. We don't need any information from them at all. And I was like, what do you mean? And they said, oh, it's like concrete. So if you remember, I said we used the syringe to make the little 1 gram aliquots. Well, the Parkinson's stool was so hard, it was like concrete, and it would not go through the syringe. They actually had to make those little aliquots using a caulking gun so that they could apply more force. So, and this ultimately has led to some research. I actually have a paper in review right now that's kind of one of the foundational pieces of kind of what led us to connect many different dots in Parkinson's. So what we found was there's a lot about in the poop of information. I mean, I'm actually just finishing writing a really short book called In Search of the Perfect Poop. And it's talking a bit about this sample collection. We had somebody who went on an all milk diet for 2 or 3 months before they sent us a sample, and their stool was white, which, I guess makes sense, but kind of crazy because never heard that before. We had a young man who was autistic, and his sample came in and it was full of cherry pits. And his mother said, oh, he eats cherries all the time, pits and all. And I started looking into that because, often the body is like giving you a signal of something that it needs. And cherry pits, like apricots, contain amygdalin that makes that cyanide compound that can also make the sulfur compounds that are in our tears or different things that are antibacterial. So do I know for sure that's what was going on there? I don't, but there were a lot of really interesting clues, I would say, or questions that arose out of that, some of which people are answering today and some of which are still out there to be answered.
Yeah. I mean, any good holistic health professional, it's not the most fun question to ask and to answer, but like the stool is a really important source of information, understand what's going on in the body. And there's other ones too, but that's one that gives a lot of information and is something that is free. So it's worth looking into.
It was interesting to me when I first started the company. I was out on a hike and I stopped for lunch, and this couple sat down next to me and they had a dog, and they were asking what I did. And I said, well, I have this company, we collect poop, this is what, blah, blah, blah. And they looked at me and they said, oh, that makes perfect sense, because that's how we know if our dog is healthy. We look at his poop. Is he pooping every day? What does it look like? All that. And it's so logical for a pet owner to think about that, but they don't really apply it to themselves. It's kind of nuts. And then in most cases, if you're just going in to your GP or whatever for a regular checkup, they're not going to ask you about your stool. And even if they do, they'll often say, like if somebody says, well, I only go every 3 or 4 days, they'll say that's normal. I'll say, normal in today's world is not optimal. And since you talk about longevity, optimal is really getting rid of that waste every day, because waste sitting in your body is producing toxins. I know we're going to talk about endotoxin, but that is one of those things where waste sitting in your body is multiplying bacteria that can produce toxins that can cross those barriers in your gut, get into your bloodstream, and be inflammatory really anywhere in your body.
So to connect the dots that we've touched on so far, you mentioned diet and understanding the food supply and even the epigenetic effect of those foods on the body, and also can these things that we're engineering, these microbes that we're engineering, can they cross over into the human host? What did you find there? And is there any link between that and this?
Well, so I did find some interesting things there. So when we did our sequencing of samples, we did what's called whole genome sequencing. So that means we got everything in the sample, like every gene of every bacteria. It's a tremendous amount of data for every sample, because in a stool sample, it's about 300 times more genes than the human genome, just to give you some perspective. So you can get antibiotic resistance genes, all these different things. And so I was giving a talk about some of this, and so I started combing through some of our data related to Parkinson's, and I had a paper on what are called marker genes that are used in genetic engineering. So, for example, often antibiotics are used in genetic engineering, where they'll take the gene segment they want to engineer into a plant, and they will blanket that segment on each side with a particular antibiotic resistance gene, so that after they do the gene modification, then they apply that antibiotic, and the only thing that survives is the genetic material that had the antibiotic resistance. And so I had this question like, okay, you're putting that into the food, we're eating the food, could that be in our gut? And so we did this analytics, and I took several of the genes from that marker gene study and showed a difference in the Parkinson's gut and controls, that these genes were more prevalent in the Parkinson's gut. Now, I don't think anybody's ever taken that and run with it to look at the research, but it certainly showed me that the genes were present in the microbiome. And then I think I found a study, it's been a while since I've looked, where they actually showed that those genes could transfer into the somatic cells or the epithelial cells in the gut. But it's been a long time since I've looked at that. So that was kind of a big part of that. And this was a multi-part question, so you might have to...
Well, is that phenomenon of the genes transferring called horizontal gene transfer?
It is called horizontal gene transfer. And when I first started looking at the food supply and horizontal gene transfer, so that was like the early 2000s, everywhere I looked is like, oh, there's no such thing as horizontal gene transfer, which is kind of ridiculous because I think there's a scientist named Barbara McClintock who had been doing research and publishing on this. And I think she had published quite a bit before 2002, but it, wasn't widely known, I guess, or it was perhaps suppressed. But horizontal gene transfer is widely known now to be the case. And the way it moves is through the microbes. And if you just think about it, the microbes are in the soil, they're on the plants, they're in the animals, they're in us, and we're sharing all that. And genes are just information, and we're all made of the same information.
Yeah. And if, as you mentioned, we're 2/3 or more microbes, then it begs the question, why are we not focused more on the microbial nature of us versus our hard-coded human genome?
Yes, that's a question I still ask every day.
And why is that? Is it just too hard to study? Why do we not know nearly as much as we should? Because people have heard about the gut being the second brain for a long time now, for over a decade, if people in the alternative health world. Why is there still not as much knowledge there as there should be?
Well, I think there's there's more than a couple of reasons for that, but a few I'll focus on is the complexity of ecosystems. Ecosystems are not simple. If you look at a coral reef and it may look like it's healthy, but some tiny little thing that dies off out of the reef is what collapses the whole reef. And so there are scientists who are looking at ecosystem dynamics in the context of the microbiome, but that's a small niche. And funding is part of that. But one of the things I would say from the early part, so I founded the BioCollective in 2015, and that's when the microbiome was really emerging in the scientific consciousness, because they had just finished the Human Genome Project, which was a 5-year massive undertaking of mapping the microbiomes of all the surfaces in different places in the body and working with all the national labs and the big universities. And so, it was this area of excitement. And going to the conferences, there were a lot of pharmaceutical companies there who were really interested, I think, in the promise of what the microbiome might hold. But as I saw it evolve over time, I think the light bulb comes on is like, oh, wait a minute. People are 2/3 microbial, and we are making all of these drugs that have never been evaluated for safety on the microbiome. Whoa, let's back up and let's move away from this, because we don't want to have to go back and look at all of the safety profiles of all of our drugs and their impact on the microbiome. And we don't want to have to look at the microbiome's impact on the efficacy of our drugs. Because there has been some work in recent years on the impact of the microbiome on certain cancer drugs. So some microbiome profiles can make the cancer drugs more optimal, and others can turn it off. So it's not simple, shall we say. Ecosystems are not simple. And so we come out of maybe 50 years of very reductionist science that wants to just kind of look through the pinhole. And you can't do that with the microbiome. You've got to have a much bigger vision and understanding about how it's all interconnected.
It seems to me if you have to rely heavily on the ecosystem nature of the microbiome, it'd be hard to use it as like a intervention target. Like, how do you say, well, I'm just going to introduce this one thing into the microbiome and all of a sudden fix all of the issues?
That's actually been one of my biggest beefs in looking at probiotics, but let me back up to my chief scientific officer who came on board in 2017. Dr. Raul Cano was a microbial ecologist, and he had spent about 35 years at Cal Poly. And he actually had an endowed chair from the oil company Unocal because he had pioneered using teams of microbes, a microbial ecosystem, to clean up an oil spill in a very sensitive area in California. And so learning from him about how microbial ecosystems work together and how the systems are working in our body, because the waste of one microbe is the input of another microbe. And so you can build these teams, and that's ultimately what we ended up doing on both the human side and the soil side. So was to build teams that put back functions that work. So if you think of it like a factory, I mean, we have different operations in our gut and different shifts, if you will, that, if you're going to build a car, you need people with different types of skills on the line. And the same thing in your microbiome. You need, organisms that break down sugars and fats and lipids and can degrade or detoxify things. And, the more toxic burden we are exposed to, the more we might have microbes that can break down those toxins, but they actually might produce some not so great stuff in that breakdown. So you kind of have to look at that whole picture. And so we started to build teams on the human side, but then Raul and I had this discussion. It's like, okay, well, you're kind of pushing a string up a hill back to that food supply, all the chemicals we're using in the food supply, how we're growing our food, the decrease in the nutrient density and all of that. If we don't go all the way back downstream and try to look at and solve the problem at the level of the soil and what we've done to the soil, which is, quite frankly, similar to Dr. Blazer's missing microbes. We have missing microbes in the soil too, because of all the chemicals and antibiotic type things that we've put on the soil. And so we started kind of thinking about it like he'd done with oil spills, but looking at, okay, what needs to be broken down in the soil to restore the environment so that we have soil respiration and we restore the carbon cycle, the sulfur cycle, the nitrogen cycle, like all of those natural cycles can come back into balance and make more nutritious food, increase the yield, and go all the way downstream to the human, and then putting back a system of microbes, which, our best-selling product, Sugar Shift, is the one I made for my husband. And that kind of came out of a conference I had gone to where they were talking about the sugar alcohol mannitol being able to stop the aggregation of the proteins in an animal model and actually pull them out of the brain. And I was like, wow, that's really interesting. And I came back and bought a little mannitol chemistry book. And the first chapter in the book was the microbes that convert glucose and fructose into mannitol. And so we started looking at that, and I was like, well, could we put a system, a team back together in the gut that can essentially convert these excess sugars that we have so much of in our diet into something like mannitol, which we essentially pee out, but on the way out, it actually feeds a healthier profile of gut microbiome because it's prebiotic. So while it might seem complicated, if you start to think in ecosystem dynamics and like building teams, it's not that different than, building a good corporate team or building a good team in a factory.
So using that as an example, how did you think through the communal dynamics and the ecosystem to make sure you weren't just going to make this one change over here and think it had this one outcome difference when really you changed all kinds of other stuff in between the upstream and the downstream?
Say for the initial product, I can't take credit for this magic view. It ultimately evolved, but our original thought process was just building a team that worked together to support a sustained conversion. And then in the process of that, we began to learn more and more how that sustained system worked together. And then from that, our bioinformatics person, who was both, biology background and systems technology, kind of put that together, and we built something called a bioflux metabolic model, where we would take the actual full genome of each of the strains of bacteria and run it through this program that could take days to run because of the data volume. And it's all built on a database from Argonne National Lab that was 17,000 different metabolites from bacteria from lab data that basically shows, based on microbes together, microbes separately, what the different metabolites are, how that all interacts over in what's called a flux balance model, which is constantly going on inside our system. And so we were looking at that flux balance, and can we build a team with this flux balance that can produce what we want it to produce for a sustained period of time. So for our sugar shift product, that's about 12 hours. And then we started to do, laboratory data, looking at all of the different metabolites and things we wanted it to produce, whether that's mannitol or butyrate, which feeds the lining of the gut, some of those things, and seeing, did it perform better as a team compared to each of the individual strains. And that's actually, we ended up getting a patent for it because the team together performs superior to any of those individual strains. And so then, taking that concept, we went on to build a product for sleep that helps support tryptophan and bacterial melatonin production, which we have in our gut, but it's disrupted by all this, you name it, from light to the food to whatever. That system can go back in and help you do that. We have one for immunity. We have one for restoring after you take antibiotics. We have a new one for stress and anxiety that really just kind of takes the edge off. And you can notice that in about 20 minutes. And what it's doing is supporting GABA production in the gut, and a lot of people don't realize, like serotonin, GABA, these things are produced in the gut. I think it's 80 to 90% of our serotonin is actually produced in our gut. And one of the early kind of fields of research I was looking at was the work of this guy named Mark Lyte, Lyte, and he was the father of what's called microbial endocrinology. And, we think of endocrine hormones coming out of the organs in our body, but essentially what he showed was many of these hormones are actually made by microbes, and there's kind of a loop going on between the organs in the body of the production of different types of hormones. And in particular, the stress hormones are very kind of gut-brain driving behaviors and choices that we make.
Yeah. Can the hormones and the neurotransmitters, the brain chemicals produced in the gut, such as GABA or serotonin, actually make their way across the blood-brain barrier and into the brain?
So they're going up the vagus. The vagus nerve is is basically the superhighway between the gut and the brain. So that's where a lot of that communication is going on. And it was interesting too. It's probably been a decade, but they went back looking at old data of people who had gotten what's called a vagotomy, where they cut the vagus nerve, and they had less Parkinson's. So they're still not 100%, but there is a researcher, I think, at Johns Hopkins, who showed that the alpha-synuclein, that's the protein involved in Parkinson's, could travel up that vagus nerve and into the brain. So there's a lot more to that superhighway, I think, to be explored. But we see now a lot of companies coming on the scene and people talking about vagal tone and dealing with the vagus nerve. And that is, it's tied to every organ in the body. And that's where that sensing is really going on, and a lot of connection to our autonomic nervous system too.
Yeah, I was going to say, because the gut is also called the enteric nervous system, the ENS, right? And then there's probably a pretty strong relationship with the microbiome in the gut and the function of the gut, as well as the activity of the vagus nerve.
So, yes, and one of the things in our paper that's under review right now is looking at the battle in the gut and how that impacts peristalsis, which is the movement of the GI tract, which connects to the vagus nerve too. But in this battle in the gut, you have gram-negative bacteria and gram-positive bacteria, and a number of these gram-negative bacteria produce something called urease, and that will kill the good bacteria. The good bacteria will spill out their contents, which is mostly potassium and a little bit of calcium. So that a tiny amount of calcium can actually cause that concrete stool. And the potassium outside the cell, potassium is supposed to be inside the cell. So when you spill a lot of potassium into the system, you basically lose that gradient for signal propagation that comes from the sodium. So it's supposed to be sodium outside the cell, potassium inside the cell, and that's where your signal propagation comes. And so that's also messing up the signal to the vagus nerve and essentially all the nerves.
So take a step back. It sounds like your general formulation process when you're like trying to develop a new product is to think about the goal you want to accomplish, then like determine if there's any known mechanisms of how to get there, and then start designing the ideal team, the community that's going to modulate, it's going to alter the existing community in the beneficial direction towards that hypothesis. And then after that, you put everything into a super complex model, and you run some like a bunch of simulations through that to figure out like what are the actual like things that are going on here, what are the metabolites that are created, how are these influencing each other, and then as a result of that, you tweak whatever you need to tweak as necessary. And then to contrast that against like what the traditional probiotics company does, they put one strain inside their product and they crank up the colony forming units, the CFUs really, really high, and then just put in a label that that's what it is and it does its thing.
That might be a little bit oversimplified. So you will find some products like that where they've just taken a single strain and done a high, what's called a colony forming unit count. So that's the number of live bacteria. You'll find some people that just throw everything but the kitchen sink in there. And we did some analytics on that and saw that, often when you do that, the dynamics of the system is chaotic. And there's not a lot of diversity in the market. So there's about, and I mean, this is a rough estimate, but there's roughly about 12 common strains that you will find in most probiotics. And those 12 strains are produced, probably 90% of the market is produced by just 3 suppliers. So if you're to go to a grocery store or a health food store or something, and they have a wall of probiotics, most of those are going to be pretty similar to each other. So you'll have your low end, low dollar that might be in the, $9-10 range. Those are going to be low CFU counts, 1 or 2 strains maybe. You'll have your higher end products that might be in the refrigeration section in some kind of a liquid formula or something in that section. But many of those are still going to have the same strains. And then you'll have some that have taken a strain that has a fair amount of research around it. So let's say Lactobacillus rhamnosus. There's and I can't remember if LGG is a rhamnosus, but I think it is. That's a very well-researched strain. And so they can make a lot of claims about it based on the research from that particular strain. So if you're looking for just that particular strain, I mean, you might want to try that and you might have some success. But I have talked to a lot of people who have said, well, I've tried probiotics and they don't work. And I'm like, well, and I try to explain, it's because you really need a team to address what your issue is. Like a single strain of bacteria is probably not going to do that. And, one of the, I'll say more on the forefront strains that's being marketed pretty heavily right now is a single strain called Akkermansia mucinophila. And I have people ask me about that a lot. It is an interesting strain because it is involved with mucin in the gut. But if you don't have a healthy gut, it can actually eat your mucin. And in people with Parkinson's and multiple sclerosis, it's actually quite elevated above the normal range. And so when I have people who say, oh, I just saw this new strain that says it's like the answer to everything in your gut. And I'm like, maybe not so much, especially if you're at risk of one of these things. You might want to be very clear that you have a very healthy gut lining and you've done some things to address the lining of your gut first before you go down a path like that. And that's maybe something we should talk about is the things that are damaging our gut lining.
Yeah, I was going to say that's an important part because I think even if like your gut lining is already okay, you're not going to hurt yourself by like supporting it in certain ways. Perhaps you do overly aggressive things, but in general, it seems like you're not going to necessarily know how thick and how resilient the lining of your gut is. So if you don't know that, perhaps work on the gut lining before using high dose single strain or certain probiotics.
One of the things I have been, I'll say, blessed to be a part of in this paper I've been working on. I've been working with Dr. Barry Ninham of Australia National University for the last 3 years. He's a world-renowned colloid and surface chemist, background in physics and mathematics. He founded the Department of Applied Mathematics at Australia National University, and somebody would go like, what does that all have to do with this? Well, I actually got introduced to him by a respiratory therapist who followed me on LinkedIn, and he said, I think you need to meet Dr. Ninham. And he started sending me his papers, and I started sending him Parkinson's papers, and could see like how these things were interconnected and explained. Of course, our gut lining is a surface, and he's a world-renowned surface chemist. Our cells, the cell wall is a surface. And what I learned from him is about something called the glycocalyx, which I'm not sure I had ever heard of it. But over the last year and a half, have really been immersed in the glycocalyx. So the early research on that was that it was the lining of our blood vessels. So it's this fuzzy hair-like lining on the blood vessels that kind of sways like seaweed and keeps things going in and out of the blood. But it's actually a lot more than that. So it lines everything in the body. It lines every cell. Bacteria have a glycocalyx. It lines our gut lining. It lines the blood-brain barrier. All of our organs, everything is surrounded by this gel-like glycocalyx. And it is made, so you have the glycocalyx, which is about 50 nanometers thick, and then you have the seaweed, the endothelial surface layer that's anywhere from 50 to 1,000 nanometers thick. So, big difference in size. And then our cell membrane is only 2 to 3 nanometers thick. So there's a lot of things that have been attributed to going on in the cell membrane that aren't physically possible in the cell membrane. They're going on in the glycocalyx endothelial surface layer. But so many of these chemicals and drugs have different interactions in the glycocalyx. If you remember, I talked about the potassium spilling into the gut. Potassium and sodium have very different impacts on how those little seaweeds stretch out and whether or not they let things in or keep things out. And so this lack of understanding about how important that is. I mean, we hear people talking about leaky gut, and I think that's been great, but it's taken about a decade for, some people in the medical world to actually agree that there is such a thing as a leaky gut. And that is basically a leaky glycocalyx. And there are many factors that are damaging that, but one of the big ones, and we're doing a publication on that as well, is a lot of our household cleaning things. So, and what happened during COVID is we just about cleaned ourselves to death. So if I go back to the early microbiome data, they were talking about the hygiene hypothesis and how, like, growing up in a really clean environment now, we're not exposed to as many microbes and all of this that train our immune system. And that makes a lot of sense. But another piece of that puzzle that's missing that makes a lot of sense is that all these cleaning products are very damaging to that glycocalyx endothelial surface layer. And that is really the sensing intelligence of every organ, every cell in the body. And so that's what's so important to support that glycocalyx. And it is made out of hyaluronic acid, which doesn't sound like a fiber, but it's actually a fiber that comes from animals, and sulfated polymers. So keratin sulfate, heparin sulfate, chondroitin sulfate, and depending on what tissues in the body, the consistency of those little kind of spiraling seaweed-like threads can have a slightly different makeup.
So if people want to work on their glycocalyx, it sounds like one of the important things to do here is to audit the chemicals you're using in the house. These are probably like the personal care products, and then also like the household cleaning stuff for like laundry, countertops, that kind of stuff. Are there any ingredients? I mean, I guess if you just choose a natural, organic, whatever, like buzzwords, a clean product, it's probably gonna be better, but are there any things to look out for?
Anything. So many of them are what are called quaternary ammonium compounds. So if they have onium at the end of any of the ingredients in them, they are very likely one of these compounds that you want to stay away from. I have noticed a few recently, like somebody sent me this one that was supposed to be really clean, but they had listed the ingredient as like C16 something something. Well, the double-chained C16s are actually some of the worst ones. So, and what happens is they can incorporate into that membrane, and they change the structure of the membrane, and it takes about 9 months for them to leave your body. So if you keep exposing yourself over and over again, then it's becoming cumulative.
Do you have any guides or recommendations there that people can look for?
So I'm actually working on a guide, and in January, so we have we do these the second and fourth Thursday of the month. We do like a little educational call-in thing, and the one that I'm doing on the second Tuesday in January is going to be specifically about this and providing a guide that people can take with them of here's what to look for, here's some products that look like they're pretty good. And if you want to make your own household cleaners, here's a recipe. Not everybody wants to do that. I totally get it. But I've even just started in my laundry, just doing my laundry with vinegar or like white vinegar or borax. 20-multein borax.
Got it. Okay, so that's the first step, is to remove the things that are interfering with and or breaking down the glycocalyx. If we want to support it in other ways, are there things that we should be adding or other things to remove?
Well, I said hyaluronic acid. Hyaluronic acid is very important to that. So, somebody who's a vegetarian is not going to be getting hyaluronic acid. So, if they're open to taking a hyaluronic acid supplement, that's probably a good idea. But, bone broths will have more of that hyaluronic acid in it. Seaweed. So your seaweeds, it's that sulfated polymer. So, and that, I've thought about that too, about the health of the Japanese, that we're always hearing that they live longer. And I think maybe there's, some connection to the amount of seaweed that they eat. So it has fucoidan also, which I think is a seaweed polyphenol. You want polyphenols. Also, there's a product that we're going to do a small study together using our Sugar Shift probiotic, which produces an exopolysaccharide that's kind of like mucus. And the Revasca product, which is designed to feed the glycocalyx. And we have a few people who have been trying it so far and having some interesting results, but we're going to do a little 30-person study starting probably sometime in the first quarter of, looking to see how the company that makes Revasca actually has a camera. It's called the GlycoCheck. And you can, if you find somebody, or, we could have people contact me if they want. I can probably put them in touch with a place, or they could go to the glycocalyx.com and see if they could point them to a place where they could get tested. But you put the camera under your tongue. So if you look under your tongue, that's probably the one place where you can see all the different size blood vessels. There's big blood vessels, small and tiny microvasculature. And the camera is taking an image of all that microvasculature and scoring the health of your glycocalyx. And so that's pretty interesting. And we had them come to a Parkinson's conference I had in May, and I scored one of the best scores. I was pretty excited because I had been taking the Revasca product for 5 or 6 months. And of course, I've been on my Sugar Shift product, since I started making it in 2017. So that probably helped, because also Sugar Shift in our clinical trial, one of the markers that so we did our trial in diabetes, and most people think, okay, you're looking at blood sugar, you're looking at HbA1c, triglycerides, things like that, HOMA-IR, insulin. We looked at all of that, and there were statistically significant benefits in all of those measures. But one of the things I wanted to look at was serum endotoxin, which a lot of people will never have heard of. But serum endotoxin is that lipopolysaccharide. It's the cell wall of those gram-negative bacteria, the bad guys, that cross that weakened barrier and get into your bloodstream and raise your serum endotoxin, or serum LPS, that is inflammatory, and it can go anywhere in your body. And that was one of the markers in our clinical trial that dropped the most. And those little components of the gram-negative bacteria, they can damage the glycocalyx. So they're one of the ways that the glycocalyx gets damaged.
Yeah, I think lipopolysaccharides is highly correlated with a lot of disease, and if not highly correlated, directly causative of a lot of conditions. So that's interesting. Why would you imagine that the Sugar Shift product worked on LPS specifically?
We knew it was very likely going to change the aerobic environment of the gut. So the gut is supposed to be anaerobic, not supposed to be air in there. And a lot of those pathogens that produce endotoxin or are gram-negative, they are what are called aerotolerant. And so I mean, partly it was just an intuitive thing for me. I knew I had studied some papers and found a researcher who was proposing that endotoxin was a cause of Parkinson's. And I looked and saw that there were animal models in diabetes using endotoxin. And I was like, okay, well, we're not going to do a trial in Parkinson's because there are no agreed upon endpoints that would be useful. But we know there's this connection to endotoxins. So let's see if we can move endotoxin. And we did. And so then I started looking even deeper. And just like you said, it's pretty crazy if you go into PubMed and you just type any disease at all that has inflammation. It could be Alzheimer's or MS or Parkinson's or diabetes. Depression is a big one. They all have these animal models using endotoxin. Autism. So I'm like, okay, like crazy. We have all these models. Why are we not asking how can we deal with the endotoxin in our internal ecosystem? It's kind of wacky to me. So that's how the idea came. And I just think I got lucky.
One of the things that always troubles me when I'm choosing a product is like, a lot of times you have to decide whether you want to take something that's going to work on like the human genome level or the microbial level. And I'm like, I could either take something that feels like it's directly interacting with me and therefore going to be more significant, as I put in air quotes, like it feels that way, versus targeting my microbiome. And like I can use a glucose disposal agent and blood sugar support that is like working on the human level, if you will, versus targeting my gut. What's the benefit of targeting the microbiome instead?
You're 2/3 microbial to start with. So when you say you could use something, a glucose disposal that works on the human side, give me an example of specifically what you're talking about.
I was going to say berberine, but I realized it's a bad example because it actually works on
the microbiome.
The gut. Yeah.
Because it is actually antimicrobial and I need to look. I think I looked at this a while back. I think it also has some impact on the gram-negative bacteria.
Yeah, I think it does.
But because it's antimicrobial, it's something that I'm going to say you don't want to use it all the time because of the killing nature of it. Whereas we're putting in something that is kind of self-balancing. It's almost like you're putting in a culture of good behavior. So if you had like a town that was sort of overrun by criminals and didn't have a strong police force or a culture that enforced the laws, then the crime might get out of hand. But then when you put the team back in and everybody's kind of copacetic, the bad guys, they kind of go back into the corners or they move to another town or they move on out your toilet.
Yeah. And I wonder also if it would have more of a modulating effect, meaning if like my blood sugar was already dangerously low and I took the microbiome supplement, then it's going to have less of an effect. It's not going to push me into like dangerous territory versus more of like the pharmaceutical, like, natural medicine approach. Even if it's just like a natural product still, it might like still push that too hard.
Interesting things about this mannitol pathway is that it is reversible. So the body, if you're converting that glucose and fructose, but the body says, oh, wait a minute, I need some glucose or fructose, it can convert that mannitol back. So it's a reversible pathway where, something that you're taking that's a specific target may not necessarily, it's not reversible until it's out of your system.
Interesting. Yeah. And then for things like the stress like product and the stress approach via the microbiome, like I could use that, or I could use something like a GABA, like a pharma GABA, pH GABA kind of thing, or I could use taurine or some other inhibitory supplement that is easy and fairly well understood in comparison to like modulating the gut microbiome. Why do you think that this is a better approach?
That's a good question too, but I think, the system is always wanting to be in some kind of homeostasis. So often supplements, even though they're well tolerated, they can swing the metabolic pathways from one side to another. And I learned quite a bit about that from a doctor who has a device that actually measures not only oxidative stress, but redox stress.
Oh, what device is that?
Hang on. Redox diagnostics, I think.
Nice.
It's available. He has a lot of dental offices that have it because before they do certain types of dental procedures, they like to see that there's, that's been dealt with. But I had a great example of overdoing reductive support, if you will. So I was going to visit one of the dentists who had one of these devices, and I thought he was going to remove a root canal that day. So that morning I took like 1,000 milligrams of vitamin C in a liposomal packet, and I put on a glutathione patch. So two kind of major reductive things, thinking, okay, I'm going to proactively prepare myself because I'm going to be under some oxidative stress.
Will you explain high level overview? What is reductive stress and oxidative stress? What is redox, like that?
Oxidative stress, you're having like these bursts of hydrogen peroxide or these oxidative byproducts that are maybe trying to kill something or deal with an infection or whatever. And then you have what are anti-inflammatory or these products that are designed to quench that oxygen that has kind of come in to deal with a problem. And so, there's lots of antioxidants on the market, but you can over antioxidant, which puts you into what's called reductive stress. Does that make sense?
Yeah.
Okay.
Yeah, the way I visualize it is that like their life exists in a balance between oxidation and reduction. Oxidation are a lot of the therapies and things that are important for certain functions. Reduction is the other side of that, and a lot of the antioxidants and things that we use and supplement for health purposes are reductive, and they counteract all the oxidation we're exposed to by living in modern life. And so it's possible to go too far in either direction.
Correct. And so I had my little test at the dentist's office, and I was way over in the reductive stress category, which kind of launched me into this whole another rabbit hole of looking at, like, are we in reductive stress? Is some of what's going on in something like Parkinson's, and looking at my husband, was, when he would take certain supplements, you could see these metabolic swings. And I think reductive stress, this over antioxidant, if you will, can be part of that as we're trying to, we got this seesaw where we're trying to maintain balance and a countertop full of all these different supplement choices, some of which are mega doses, and electrolytes actually being one of those that we determined was causing problems for John, because depending on how the electrolyte is formulated and what the balance of the different ions in the electrolyte are, because those different ions affect the glycocalyx and signal propagation and oxidative and reductive stress, like all of those pieces were, he was swinging pretty dramatically back and forth. So I think to sort of circle back to using microbes and a team of microbes, because they are the workhorse of the gut. Many of your vitamins, hormones, neurotransmitters are all made by those microbes. So if you can restore that healthy balance, restore those functions, and provide them with a nutrient-dense diet, which is harder than you think, then they should be able to produce most of what you need. But we're in a situation now where we have a little bit of a desert type landscape where we've had a scorched earth, if you will. And so those systems are not working that well. And so
how do you know if you need that extra support so that your internal pharmacy can produce everything that you need, or if you're already there?
that is a great question, because I'm still, I still look at people send me different microbiome tests, and they're getting better. So some of them now will show whether you have B vitamin production. Some of them, I think, can show whether you have the bacteria that are involved in vitamin K synthesis. And different tests have different pieces of it, but I would say there's nobody yet who's really doing a great comprehensive job that I've seen of kind of putting it all together. And I listened to a talk by Jill Carnahan at the GIA4M, I think back in August. And she was talking about all these, different microbiome tests and trying to look at them. And one of probably the most important thing is if you're going to start with a particular test, you should stick with that for a bit as you try to look at your microbiome, because different kits have different biases or different focuses. And so you don't want to just hop around to all the different ones, because they're impossible to compare to each other. So I don't know if I really gave you a great answer there, but it's come a long way since 2015. And when we gave this 60-page printout we had a little bit of a report who gave some people some general ideas, but the 60-page report was every bacteria, like how much there was of them, all the antibiotic resistance genes, all the virulence genes, all the fungi, it was like all this stuff, and people would take it to their doctors and they would go, like, they don't know what to do with that. They haven't been trained on that. And we're still a decade later, and functional medicine and naturopaths have been trained on looking at the microbiome. But even they're not experts. There's a few. I would say Jill Carnahan is the closest to an expert that I have seen in listening to her talk about it, who really deeply understands it. And I'm sure there's more like her, but there's an awful lot of people who hold themselves out as experts that really aren't.
Yeah. So it sounds like even if your gut you don't have any gut symptoms, you don't have any digestive complaints or anything like that, it's not necessarily indicative that your microbiome is producing all of the vitamins and chemicals that you need it to produce. And so there are tests currently, none of them are a gold standard that really will tell you everything. We'll probably get there at some point, but working with someone who really understands this landscape well right now is the best thing that we can do, in addition to a lot of the core foundational gut health, universal principles.
Exactly. Great summary.
Are there any of those universal principles that you think warrant a minute of conversation before we start to wrap up?
Yes, I would say the universal principle of being conscious of what you put in your body at the simplest level. That goes to, people often say, there's so many different things. What do I do? What's one simple thing I could do? And I'll say, the simplest thing you can do is drink clean, filtered water that has minerals in it. So if you're using reverse osmosis, you need to add back minerals because you're stripping all the minerals out. But our water, people don't understand that our water systems were designed to remove bacteria, but now they have a lot of chemical pollutants in them. Every drug your neighbor pees out goes into that wastewater treatment plant, and the systems aren't designed to take those out. So water, to me, is the most essential and simplest thing that we can do, is start to think about the quality of the water we put in our body. And then number two is the quality of the food. And not everybody can afford organic, but I do know, here at our farmers market, they take the SNAP and the government assistance they take at the farmer's market, and you can ask the farmer, like, have you used chemicals? What chemicals have you used? And they're very open and honest about what they use and when they use it, and if they need to use it, why they need to use it. So I think those are the two kind of foundational things that I tell people, because like you could go for hours on all the different things to think about, but those are the two simplest things. And they go all the way back to when John was first diagnosed, to the two things that we did, which we cleaned up our water supply and we cleaned up what we were eating, and we ate whole foods that we made ourselves in our home more than eating out in restaurants. And I think those things alone had a big impact on, unfortunately, John passed away in 2023 from a pulmonary embolism after kind of struggling with long COVID. But he had a very active life up until the point that he had COVID. He was still rock climbing, driving, running exercise programs, doing all of that, 20 years into his Parkinson's. So just those two basic things can have a huge impact on your health.
Well, Martha, you brought us back full circle. Thank you for that. If people want to connect with you to try Sugar Shift or any of your products and future inventions, how do they go about finding you?
So you can find me on our BiotiQuest website, and that's BiotiQuest.com.
Perfect. And you already gave the final words, so thank you for joining me today on the podcast. It's been a pleasure chatting with you and learning about this interesting and complex and still not fully understood world of microbiome science and how that connects to the outside world.
Thanks so much for having me today, Nick. I really enjoyed it.
Me too. Bye, everybody.
Thanks for tuning in to High Performance Longevity. If you got value today, the best way to support the show is to leave a review or share it with someone who's ready to upgrade their health span. You can find all the episodes, show notes, and resources mentioned at outlier.com. Until next time, stay energized, stay bioharmonized, and be an outlier.